So I have to share the things. So hopefully not too difficult. Okay, so regarding the topic, I will first introduce shortly Oomycetes, and after that I will go through the different lineages and tell a bit about the differences we can find there. So Oomycetes are causing billions of damage every year in agriculture, forestry, and aquaculture. But some also threaten the native species, for example, the great thing. However, when people were first looking into this, it was often not really well understood what is happening there, because the organisms causing the diseases are really very significant. And in the middle of the 19th century, microscopy became very established, so people saw really very weird things that they tried to explain based on what they knew at that time. For example, here, this is something that Nägeli saw. He saw an endogenous cell formation in fungal filaments of a mollusk parasite, and this was in 1844. And he was not really sure what he was seeing there. And various scientists found this is pretty interesting. And about 11 years later, Brown, we already had introduced in a previous talk, understood that these are actually parasitic things that are growing inside a fungal filament. And a bit later, Cornu noticed that this parasite was something really different than everything else known so far. And the first Oomycete was described as Olpidiopsis subcolegniae. Holocarpic Oomycetes, in difference to the true fungi or to many other Oomycetes, are taking everything that they have in their thallus, in their sporangium, and convert it into zoospores. So where can you find Oomycetes? So actually, Oomycetes can be found here in Stechlin, but it can also be found in the Mediterranean Sea, in deserts, in the Himalaya, in mangroves, in rainforests, savannas, oceans, and also in the Arctic. So Oomycetes can actually be found pretty much everywhere. Okay, so if we look into Oomycetes on the tree of life, so what we had before were the fungi. The fungi you can find there on the left side next to the animals, and so they are our sister group, so to say. And the Oomycetes, they are belonging to the Stramnipila, which is something completely different. So you can see them on the right side here in this tree. The Stramnipila are related, for example, to alveolates, where we have the dinoflagellates and epicomplexans, such as malaria. And the Stramnipila are characterized by having a tinsel flagellum. This is a flagellum that has multiple hairs on. You can see it in the upper right. And those hairs, they are reversing the thrust. So which means if you can see an oospore swimming that has a flagellum directed forwards, then you already know, oh, this is pretty likely an Oomycete. So within the Stramnipila, we have lots of different organisms. The most well-known probably are the brown algae that can form the kelp forests, but then also the diatoms and chrysophytes belong to this group. And then in the bottom right, we have the Oomycetes that have those nice oospores forming in the more advanced groups. And the Oomycetes, they have a quite diverse set of lifestyles. They can be saprotrophs, like in Achlyea. They can be parasites of diatoms, like in Nisma. They can also parasitize other Oomycetes. They can be endophytic in plants, and they can also cause various plant diseases. But today the talk will be only about those groups that are obligate parasites, and they are parasites of different algae. And the parasitism on algae or diatoms has been arising several times in the evolutionary history. So basically throughout everything, you can see here that there are different lineages that have developed those lifestyles, where you can see a small thallus inside a cell, and all this thallus will later convert into zoospores. So the holocarpic Oomycetes are thus spread throughout the whole lineage of Oomycetes. Within the Paranospiralis, we have one eukaryotic species, at least one, which parasitizes algae, in this case Porphyra and Pyropia, and this is Pythium porphyri. And then we have some additional ones in there. Pythium porphyri is causing great harm to algal farming in the Pacific, but also increasingly in other areas where porphyri. Then another group also within the Paranospiralis is Laginidium nodosum. Laginidium nodosum is causing the decline of cyanobacteria, especially of Lyngbia and Oscillatoria species. And as you can see here, the parasite, when it starts out, it is in the upper panel. You can see there is a small vacuole-like structure, and you can see a hyphae growing. And if this is successful, those first stages, then you can see that the hyphae will grow further. You have these nodulate structures, which gave rise to the name nodosum. And if everything is successful, then you can see in the lowest pictures, there are zoospores formed, and those zoospores, the spores, they are formed inside the vesicle and then released after some time when the vesicle dissolves. But then also pretty news, we haven't published this yet, but we have submitted a manuscript on this. There are also those Lagena species in diatoms that also belong to the Paranospiralis. And also this group is pretty diverse. Actually, we were very much surprised because there was not a single species of this group reported so far. And yeah, but we found that there are at least 4 species that we could identify already, and we expected there will be many more. So this is a pretty unusual group. Then if we move to the second group of Oomycetes, the Saprolegniales, here we are also having some parasites of diatoms. And in contrast to the previous group, to the Paranospiralis, in the Saprolegniales, 2 different generations of zoospores are formed. So which means they have a first generation of zoospores that are usually weak swimmers or sometimes just cysts. And then the second one that can swim very nicely and vigorously. And what you can see here is Aphanomycopsis. This is Aphanomycopsis bacillariacearum. It can be found on Pinularia. And in this case, there are cysts formed, and from those cysts, you will later see the zoospores hatching. And if they are empty, you can still see there's a bunch of those cysts at the mouth of the discharge tube left. Okay, then we are leaving the Saprolegniales. We are going to the second group within the Saprolegniomycetes, which is the Leptomytales. The Leptomytales have previously been also called the early diverging Saprolegniomycetes. They are a group that is very diverse and very ill-characterized, so we don't really know much about those. But they contain one of the most important oomycete parasites of marine algae, which is Lagenisma costinodisci. And this is parasitic to the large centric diatom Costinodiscus, and it can form hundreds to thousands of spores from one diatom, so which means it leads to quick decline also if you have them in culture. And we will also see them in the afternoon. So Anthony, who is bringing some cultures of Costinodisci, and then you can probably also see how they release all the zoospores. These groups cannot be cultivated, so no successful cultivational media so far. This is also basically the same to all the other groups, except for Pythium on the red algae and for Laginidium nodosum on the cyanobacteria. Also in the Leptomytales, there is the second big group, that is Ectrogella. Ectrogella was basically the dumping ground for all oomycete parasites parasitizing diatoms. And of course, it's way more diverse. So we now know that the oomycetes infecting diatoms are very polyphyletic, 6 different groups. And Ectrogella infects large pennate freshwater diatoms like Nichia. And there are many species in the genus, but probably just a very few will really belong there. Okay, then we are now leaving the crown group. So within the Oomycetes, about 95% of the species are actually described in the crown groups, which is the Peronospiralis, Saprolegnalis, and Leptometalis. And then there are many, many basal groups that are not really well known. And one of this is the Protismatalis. And Protisma, the genus, has been thought to include only parasites of red algae, but actually it also includes parasites of green algae that were formerly known as Xeropedium. Also, this group cannot be cultured so far. And it's also maybe noteworthy that many of the red algal parasites has been previously assigned to Oomycetes, but Oomycetes is something completely different. I will come to this later. Then another group also in the early diverging Oomycetes are the Anisopiliales. The Anisopiliales are containing two genera that are parasites of marine algae, and Anisolpidium, for at least the species published so far, they are infecting filamentous algae, and characterized by having no whiplash flagellum, but just the tinsel flagellum. So which means they are one flagellum that is directed forward and not a second one that they are dragging behind. And also those ones haven't been cultured so far. The second group in the Anisopiliales is the genus Diatomophthora that has been pretty recently described. And also because this is not at all related to Ectrogella. Ectrogella is in the crown Oomycetes and Diatomophthora is in the basal clades. Diatomophthora infects a wide range of marine diatoms. For example, here you can see it in Rhizoscelenia in the lower panel, and also limnic diatoms in the upper panel. You can see some Pleurosigma, and they are forming zoospores that will be swarming already within the sporangium, and then they are released through a short discharge tube. Probably noteworthy is Diatomophthora perforans, because this is an exception actually, because it infects very, very many different hosts. You can see it also this afternoon in Costinodiscus granii, where it's infective, but it can also infect pennate diatoms actually. So it's also infecting Ulnaria species, and also it's infecting some, basically almost everything. So we have tried to put it to several different marine diatoms, and it can take almost all of them. Okay, now the short excursion to Olpidiopsis. Olpidiopsis is really confined just to parasites of Oomycetes, as far as we know so far. What you can see here is Olpidiopsis saprolegnae infecting a hyphae of Saprolegnia. And if it infects the hyphae, they are becoming hypertrophied, and then they are forming the sporangium inside. And they are pretty basal in the Oomycetes, but not at all related to the other groups that had been previously assigned to Olpidiopsis, which are the red algal parasites there with the red brownish shading, and beneath with the yellow brownish shading. These are parasites of diatoms that also had been previously assigned to Olpidiopsis. But Olpidiopsis, as it stands, is really only restricted to Oomycetes, at least the confirmed species only in Oomycetes. Okay, then we go further down the tree of life, and we are coming to Eurychasma. Eurychasma is a genus that infects brown algae, and the interesting thing in this species is that it also causes hypertrophy. You can see there in the lower panel, you can see an infected host cell, and it becomes really large by this. And then in the interior, you can see that there are small beak-comb-like structures on the surface. And this is typical for Eurychasma, that you have this net-like or beak-comb, honeycomb-like appearance within the sporangium, which is the place where cysts are attached tightly to the sporangium wall, and they will then give rise to zoospores that escape through the exit tubes. And then last but not least, probably most basic group, the Oomycetes, the Miraculalis, so far with just a single genus, the genus Miracula, has been also pretty recently discovered from a toxic marine genus, which is Pseudonychia, and it's really very early divergent. And over the past 5 years, we've been looking more closely into parasites of diatoms, and we have found some new species, and this species also seems to be pretty widespread. It occurs in both marine and limnic diatoms, and it's also occurring both in centric and annate species. But it seems to have a pretty high host specificity. So usually when we find a species of Miracula in a certain diatom, we do not see infections in any other diatom. So also this genus seems to be a pretty diverse one. And then if we are coming, slide is not there. When we are coming to the unknowns that you already heard before, this is just like one experiment that we did there in for Oomycetes, and there you can already see that in both the Miracula clade, especially in the Diatomophthora clade that was formerly assigned to Epidiopsis, we see lots of OTUs that are very highly distinct and are representing species that are to be discovered yet. So it is really likely that those maybe 20 or so species that we know of the diatom parasites so far, that this is really just the tip of the iceberg, and we expect that there will be probably even hundreds of different species, judging from the host specificity that we see for most of those parasites. All right, so, and with this, I'm already summing up. And yeah, it's noteworthy that you have really lots of interesting reports in the old literature, many of which are not resolved so far. And it has been doubted sometimes, but I'm pretty confident that these reports refer to real organisms, and that also the minor differences that were sometimes found are probably behind a much bigger diversity than has been anticipated. The holycarpic parasites of Oomycetes, they're really everywhere, and they play a major ecological role, which we really don't know very detailed so far. As mentioned, for this marine-centric diatom, Costinodiscus, this is responsible, the genus itself is responsible for several % of the global photosynthesis, and the Oomycetes, because they're producing so many spores, they are really able to break down such blooms. So it might be that this is also a group that has been pretty much underestimated in terms of the ecological significance. The holocarpic Oomycetes are not a part of the genus group, and parasitism on diatoms and other algae has evolved multiple times independently. And yeah, most of the holocarpic Oomycetes, they are still undiscovered despite their ecological importance. And yeah, just like in the last 3 weeks or so, from another trip that we had to Iceland and isolated some holocarpic Oomycetes, I expect there will be another 5 new species that we can describe from there. So it's really a big biodiversity that is very much unknown. And I'm also confident that also in the Lake Stechlin here, there are new species to be discovered. And with this, I thank you for your attention. And yeah, I'm ready to take questions now or afterwards.
Okay, thank you very much.